Chief guest, Professor Zahid Ashraf, Dean, Faculty of Life Science, Jamia Millia Islamia. Professor Ashraf is a distinguished scientist whose pioneering work in high-altitude biology and thrombotic disorders has made significant national and international impact. An elected fellow of India's leading science and academies and a recipient of several prestigious honors, his research exemplifies translational science at its best. Now, allow me to share a short video with you all regarding his research.
Humans and almost all life forms on Earth need oxygen. Oxygen for every breath they take. Hypoxia in biology and medicine refers to condition of the body in which tissues are starved of oxygen. Professor Zahid Ashraf, working as head of the Department of Biotechnology, Jamia Millia Islamia, has focused his research that was of urgent need and challenge for nation and scientific community to elucidate the blood clotting problem inflicting our soldiers that are positioned at high altitude. The disease involves a thrombus, also known as blood clot, that forms in a vein deep in the body. Most often occurs in the deep veins of the legs or in brain. The thrombus could travel to lung and results in a fatal condition known as pulmonary thromboembolism. Professor Ashraf's research contributions have increased our understanding that includes prevalence, mechanism, genetic, diagnostic, and development of treatment against hypoxia-induced thromboembolic disorders. His research revealed a novel cause for blood clot formation at altitudes and discovered calpain. Calpain as a biomarker for early diagnosis. Further, he discovered previously unknown cause of thrombosis at altitude, and it's regulated by a complex network of coagulatory and inflammatory responses. Hypoxia-inducible factors, HIF-1 alpha, activates NLRP3 infusomes and releases cytokines leading to blood clotting. The same inflammation, coagulation, hypoxemia convolutions has also been observed in COVID-19 patients. In a rare breakthrough, his lab developed a small nucleic acid microRNAs mimic-based therapy against thrombosis. This contribution of Professor Zahid for societal benefit, especially for armed forces defending our nation.
With this, I now request Professor Muhammad Zahid Ashraf to kindly deliver his lecture on the plot behind the clot, biomarkers and breakthroughs. Sir, over to you.
Well, thank you so much for Swati for such a nice word. And I'm audible, everything perfect, audio-wise and all. Hello?
Yes, sir. Thank you for
joining. Are you able to hear me?
Yes, sir.
Okay, so you have to do me one favor. You have to allow me to screen sharing only, then I'll be able to.
Okay, sir, just a moment.
1 or 2 minutes break. I also take a coffee in between.
Sir, our technical team is working on it. Just 2
Yeah.
minutes.
Yes, sir.
So I hope it is working now. Kindly check. So your mic is off.
जी सानू सर एक सेकंड
So are you being able to share the screen now?
Sorry, just there was examination is going on, so there was some issue. So I still not able to see share your screen or something. I guess you have to make co-host or something, or I can mail you either way my PPT. In the meantime, those who are comfortable, they can interact. They can.
Yeah, I think this is at the bottom left, third icon from the left. Share screen. Do you see
So
that?
that is not enabled in my. So either I have logged in as a guest or something. You have to some kind of. So this is not a law. That is enabled. So this is not enabled. I'm not able to do this share your screen. So you have to add something, you know,
You
some
are logged in as
invite or some guest list or something.
I'm adding you as a co-host now. Is
Yeah,
that
now
okay?
it's fine. Yeah, wonderful.
Good. Thank you.
Share the entire screen. So are you able to see my slides?
Yes, sir.
Okay. So somebody has to be like, you know, in between, because the drawback of Google Meet is that I'm not able to see anything numbers. So I'm just only able to see my presentation. Are you able to see this, my PPT? It's
Yes, sir.
changing,
It's visible.
changing.
Yes, sir. It's working fine.
Okay, wonderful. Thank you. So once again, good morning and welcome to all the participants, and then sincere thanks to the organizers for inviting me, although not from the exactly field, but overall the whole idea is to, you know, how to carry forward a challenge which you are given up, and how to translate how to convert this into a translational outcome. So another 45 to 50 minutes, my point would be to connect with you, to share some of our successes, some of our, what do you say, that failures, but largely we have been successful in putting forward some good leads, which could be not only an example for the disease I work on. So what we work on is that our lab mainly focused, from the video you might have seen, is that hypoxia biology. And hypoxia per se is, I mean, there are a number of ways where, or situations where hypoxia is either in the localized form or in the systemic, means whole body is exposed to. So one of the things which we work on, I work for Jamia Millia Islamia, you see, but at the same time, my field is that I work on the mountains, and then these are the two images from, just to say, from the same place in the month of April and in the month of November. So we just have a very limited window to work on. This two of my student, there's nothing much to play around, so he was playing with a local dog. So what I'm saying is that whatever we are trying to do is a very limited time window and logistically very challenging. So coming back to hypoxia, is that we all, since most of you, I would say that from medical background or from the research background, who were working on various aspects of research, mostly in the cancer related, but hypoxia per se, everybody understands, is that the condition in which tissues are starved of oxygen. And at the same time, that historically, when the eukaryotes has generated that 450 million years ago, that body has somehow evolved the mechanism to ensure that sufficient oxygen is supplied to tissue via medium which mainly include bloods. And then this still we have what we call a hypoxic situation, either during exercise or sports activity, or during one of the other major point example of hypoxia, and which actually match with the theme of this Gyan workshop, is that cancer is a classic example of localized hypoxia, where the cells are tumor cells are under extreme hypoxic condition. We are also working on some aspect of that, but today that is not the topic for the discussion. The other example is pregnancy, womb per se. So what we say that humans' origin or genesis itself involves hypoxic environment. So we are, and you may be knowing that fetal hemoglobin has a more oxygen carrying capacity. So human's origin itself is connected with hypoxic environment. And then the last what we work on is what we call high altitude, or that the environment or the mountains where because of elevation, I'll show you in coming slides, that we are able, you have less oxygen. And this not only provides understanding the disease or the challenges too, but it provides a very beautiful example for understanding gene environment interaction, technically epigenetics. If you want to look for the human evolution or adaptation through the time scale, this provides a very beautiful natural window to work on. To get you acquainted with hypoxia or high altitude, what we call, this is what we call gas law, the PVs, NRTs, that when you go up, the pressure is low, and then barometric pressure goes down, and then accordingly the oxygen, that inspired oxygen level goes down. And then we technically divide this whole altitude into what we call high altitude, from in a general terms up to 10,000 feet. Then from 10 to 15,000 feet we call very high altitudes. And then the third is that anything beyond 15,000 feet, where the Mount Everest lies, and then where that our main that Siachen mountains lies or Himalayan range lies, is extreme altitude. Otherwise, all over globe, these are not very inhabited places. And the body responds to various physiological and adaptation in response to exposure to these altitudes. And why India has to worry about it, this is a global map of extreme altitude. And you see that there's extreme blue, if you're able to see, is that our high altitude mountain range or Himalayan range is one of the largest high altitude mountain ranges all across world. So roughly around 140 million people stay, and we have few major cities which falls in those range. India has another thing, is that what we call religious pilgrimage, which is not common in other parts of the world because of the Char Dham Yatra, Amarnath Yatra, Kailash Mansarovar Yatras. Roughly 6,000 people travel across. Other part of the world, mainly mountaineering and winter sports guys, they move to mountains, but we have this, and that lead to a lot of discomfort, a lot of diseases. Also, this is just to show you in a perspective. We have few of the major cities, and mainly Gangtok and Leh, which has a quite sizable amount of population, which is situated somewhere like Leh is somewhere, if you convert them into feet, is somewhere like 11,000 feet. Same way, Gangtok is much higher, but the advantage is that they have a lot of greeneries. We have other hill regions which are situated at altitudes. But why India has to, and why we need to learn more about high altitude, is because of Siachen Glacier Reserve. And this is a nice picture of Karakoram Range. It falls in extremely high altitude region, somewhere like around 20,000 feet. So human body has its own challenges or its own capability, physiological capability to deal with these challenges. If they are not able to deal with metabolically or with the immune response or with adaptation, mainly in the respiratory or cardiovascular system, they could lead to several dysfunctions, or several diseases like loss of RBCs or anemic condition. Cancer is another, a stroke, infarctions, and all those. So what today's talk and what we mainly focus on, adaptation, how human body is able to adopt these high altitude challenges. And one of the major thing is that if your body is not able to adapt to these challenges, it results into three major diseases, what we call acute mountain sickness. I'll not get into details of it, but if those who are interested, they can further get connected or learn about from us in any form. The other is that what we call high altitude cerebral edema, and then high altitude pulmonary edema. So technically these AMS is kind of milder form. Cerebral edema is very severe and then leads to a lot of fatality. Adaptation, high altitude pulmonary edema leads to this coughing, tachycardia, and then like fluid accumulation inside lungs. What we focus on and what today's discussion is mainly about, what we call the fourth kind of disease, what we call HAIT, high altitude induced or altitude mediated thromboembolic disorders, which mainly involves deep vein thrombosis, means vein, the blockage and the venous thrombo in the flow of blood from the lower limbs. It could result into what the situation we call, you might have seen in the video also, pulmonary thromboembolism, through which these all the clots which travel eventually from the your legs to eventually the alveoli of lungs, and eventually they obstruct the normal air passage, and which could be fatal in few times. Now the symptoms wise, or these are just too graphic to make you understand that acute mountain symptom mainly deals with the headache and nausea, all of those things. HAPE is slightly on the severe side, cough and on tachycardia, and then central synapses. The haze is that apparently it is not visible, but there are various symptoms through which that could be identified, and there is a very fatal disease. And what we, the fourth one, what we call pulmonary thromboembolism, is mainly deals with the clot formation in the lower limbs, which eventually turns to your heart and lungs, which could be fatal. So my lab works on what the thrombosis or blood clotting disorders at altitude, and especially deals with hypoxia biology. So what when we started this whole project, or when I joined almost 10, 12 years back to DRDO, what the problem which has, intrigued us is that there were anecdotal evidence, like there are much higher ratio or incidences of what we call deep vein thrombosis, which results of which is clot is being formed on the lower limbs, which eventually result of what we call PT, thromboembolism, pulmonary thromboembolism, or mesenteric, or the cerebral venous thrombosis. As a whole, the clot, both in the arterial system as well as venous system, for those who are not familiar with this, that the blood is going towards to the heart and coming out. Both the systems you eventually we looked into, that there are larger incidences or more episodes of VTE at altitude. So we looked into, like any other disease, VTE or clotting also have various, what we call acquired diseases. Anything? So acquired means that, surgery, major trauma, immobility, malignancy, long hospital admission, these all could result into some kind of clotting problems. And the same way, like any other disease, it has a various genetic factors, like, factor V Leiden, prothrombin dissolve, proteins C and S, what we call our body's anticoagulant, which keeps the body, our fluid, blood in the fluid state, deficiency in those. But with the effort, with our multiple efforts for almost a decade, what we were able to now establish hypoxia or high altitudes an independent risk factor for this disease. So that's why we have labeled them or referred them as what we call HAIT, or high altitude induced thromboembolic disorders. Our lab works on mainly, basic science and the molecular mechanism, the tools to diagnose them, and then how we can eventually get into effective management strategies. So I'll just start from the first fundamental. So when we know that high altitude has a more incidence of VTE, so we wanted to first, in a first series of experiment, to know whether this is the same thrombosis, the occurrence or the biochemistry or molecular signature is the same of the thrombosis which occurs at the plains. So what we have taken samples from 50 patient sample from Delhi, and compare that from another 50 samples of 50 patient data from the patient who has developed thrombosis while working mainly at the Siachen Glacier region. So to make the long story short, what we were able to identify is that if you compare the yellow versus the blue, that the clotting or the thrombosis at altitudes, what we call HAP, in high altitude patients happens at a much younger age or compared to the sea level patients. And then another intriguing thing which I've encircled is that it happens not only so the normal thrombosis is mainly clotting factor driven mechanism or the phenomena, but what we were able to uniquely see that it involves a lot of other vasculature which were not known for in case of especially venous thrombosis. So we said that it is not only that clotting thing protein which get triggers, there are a lot of other factors which are involved. So our journey has started with asking a very fundamental question that yes, in the normal circumstances, your optimal oxygen level, your blood cells keeps moving. But what happens in the reduced oxygen environment is that somehow some perturbance happens, and then initially the platelets get stuck to the what we call blood vessels or endothelial surface, which triggers with a series of now we know events which eventually results into clot formation and which as a whole makes blood much more thicker. So we have taken various approaches. I'll start to give you some sense. So one of the first thing what we have done to address any issue is that you should have model, either cell culture wood-based model, or you should have in silico model, or some kind of animal models or humanized models, what we call a knockout. So some for addressing cancer or anything. So we dealt upon to establishing a model for the understanding this disease, and we have asked a very fundamental question using various approaches. Gene expression analysis work, which has again showed that the expression patterns of gene. We have looked into cell biology, looking into various involvement of various immune cells or vascular cells or platelets, endothelial cells, all those. And then we have carried out extensive what we call animal model based thrombosis and coagulation based assay. And one of the key coagulation based assay now after COVID everybody knows is that D-dimer, which gives you some sense is that's yes, clotting has happened. And so in all too long, again, take home message remains with the various ways we were able to make a point that yes, oxygen deficiency per se was responsible for accelerated thrombus, or they could be a major trigger factor for the thrombus generation. So whenever you think that you are the smartest chap on earth, that's not the case. Parallelly, group from Harvard was working on the same issue, and then they have also come up with the similar finding that hypoxia encountered at altitude promotes deep vein thrombosis. They have used the mice model, and we have used parallelly the rat-based animal models and then human phenotypes. So their article got published in the month of September end, and ours is that November mid. So this is very rare, or this is a fascinating sign that parallelly group from India and group from US have been able to publish both. Their take-home message was the same, that hypoxia, in case of hypoxia, there is an increased clot formation, and then we have said that yes, bleeding time is decreased. So technically, that clotting accelerates in the hypoxic environment. The only main thing is that which you will rarely hear, that our paper get published in somewhere in the very prestigious journal Blood, which something impact factor of 20, and the Americans got their publishing impact factor 5 or 7. So which is a rare coincidence that Indians got the credit for this. So just not to praise our lab, but to inspire others that if you have a good fundamental questions. And again, the basic point was that we have done a very basic experiment, that what we call blood clotting dot experiment. What we have done, just to emphasize on that, we have taken a blood filter paper and we have just touched the filter paper with we have done a puncture in the tail of animals who have been exposed to hypoxia, and then compared with the control, and then every minute we have touched their tail to this filter paper, and you will say that even that kind of what we call 10 rupees experiment, we're able to do give us a good sense that exposure with the hypoxia suddenly results into viscous blood, which spreading is much shorter as compared to the control. So what I'm trying to highlight the point is that if you have a fundamental good question, you don't need hi-fi tools. And the so nice of blood that they're told to retain this image. You see, they have used this, Americans have used the knockout mice and then thromboelastography, which cost somewhere like 30 lakhs. At that time, we did not have those equipments, but we have the right question. So the second thing was that once we were knowing that that's a clotting is happening, much accelerated pace in exposure to hypoxia, we wanted to understand that what are the key events. And I left this all classical experiments, that one of the key experiments are that, or one of the key events are that the platelets would get activated and they will attach to the inner vascular layer, what we call endothelial surface. So that we have done all those time course and those and different experiment to make a point that platelets get hyperactivated in response to hypoxia. We wanted to see what is happening inside the platelet. So we have taken a platelet proteome approach, again to cut down to the point that we have exposed the animals to hypoxic environment, and we have isolated their platelet, and we have then the whole proteome analysis of the platelet. We were able to see somewhere like 150 different proteins are differentially regulated, and these all are what you will say that bioinformatic analysis and all the functional analysis and all. But we were clearly able to see a signature for platelet-derived proteins which are modulated in response to hypoxia. So we have looked into various proteins, and then you can. But I just want to cut the story here to one of the things what we are able to identify is that what we call calpain. Family of calpain is a protein, or what we call cysteine proteases, which is activated by calcium, and which suits very nicely with the calcium is in response to calcium release, the platelets are getting activated, and one of the calpain remains one of the key proteins which does various clippings or which initiates the process of blood clotting. And you will see this is the proteomic data, and then we were able to confirm that into animals, and we have done all those bioinformatic and then all networking analysis to make a point that they were not earlier reported to be connected with coagulation. And to get into proof of concept experiment, what we have done is that we have treated these animals with the platelet with the calpain inhibitors, and then you don't have to get into what clotting experiments we do, but just simply see that the images that animals who were pre-treated with the calpain inhibitor, which are significantly reduced blood clotting or delayed. If we looked into the phenotype, we looked into the clotting patterns, those all were significantly delayed or slow kind of. So we were able to make a point that calpain could be a leading cause, and Blood wrote an editorial on our paper saying inhibit the calpain to climb the mountain. So we were able to introduce a new player, calpain, as a family of cytosolic proteins whose enzymatic activity depends on, and then I'm not getting into a structure wise, but these all are like proteases like caspases you will hear a lot in the cancer research are active. These proteases are activated by heterodimerization. So to finally conclude this part of that, we were able to establish or set up a new area of research, and which has given us the discovery of calpain as a novel biomarker. And then our focus was to the early detection. So we were able to use this, DRDO has used this for developing kit for early detection of thromboembolism at high altitudes. Then the second question as a basic scientist was that, yes, discovery is fine, that we were able to find out calpain as a known biomarker. What are the early factors which regulates hypoxia-induced thrombosis? So for answering this question, we embarked upon what we call proteomics approach, and we like, fundamentally, if you get into central dogma, the protein, before protein, there are set of DNA and to RNA, which RNA technically regulates the expression of the protein. So we have taken what we call a snapshot of animals from the transcriptome angle. The whole idea was to look into what are the early um, genomic factor, or which are early transcriptomic signature, which could give us lead for understanding this disease in more details. Again, these all the NGS data, and finally analyze on the basis of various bioinformatic tool. I just want to make a simple point here that we looked into various cellular responses in response to hypoxia, especially in the animals who have developed thrombosis. So one of the key, or one of what we call in bioinformatics, is the nodal point, a nodal molecule turned out to be IL-1 beta. So IL-1 beta, again, that suggests that hierarchical activation of intense inflammatory response in the hypoxia-induced thrombotic animals. So what we did is that we went back to looking into how this IL-1 beta could be regulated, are regulated in the system. So those who work on, and again, you will hear about this NF-kappa B signaling pathways a lot in coming days based on the cancer. There are key molecular mechanism involved in cancer. They are equally the same. They are there in the pathogenic situation. So we wanted to look for IL-1 beta. What could be? We went back to our transcriptome data and wanted to look into what are the key factors which are actually regulating the expression of IL-1 beta, which could be technically the end productive. So one of the key thing, one of the, other than NF-kappa B signaling pathway, there's another pathway which regulates and that IL-1 beta and IL-18 expression. You just have highlighted these two cytokines. We looked into NF-kappa B signaling and we were not able to see any major change. But what we were able to see is that NLRP3 mediated, or what we call non-drive receptors being activated, which further leads to release of NF-kappa, IL-1 beta. So just to put a few words about this, all holistically, no, at that time it was not very well defined, but later it was termed as what we call inflammasome complex. So again, since audience, for the larger interest of audience, I just kept it that inflammasomes are technically what we call multimeric protein complex. And multimeric protein complex means that it has a three major components. If you look into that, in node-like receptors, which are connected to what we call adapter proteins, and one of the key adapter proteins, so NLRP is a node-like receptor, and there are various class up to 14 different, but what we focusing today and what are the majorly focused are the NLRP3, and they are connected, or they are associated with this what we call PYD domains to adapter protein ASC, and these adapter proteins convert this pro-caspase into an active caspase 1, and they sometimes, that they are connected with another protein NEC7, and then this whole ASC, NLRP3 and caspase 1 creates what we call inflammasome complex. Inflammasome complex are activated by two major pathways, what we call pathogen PAM, pathogen-induced pathogen-mediated pathways, or the DAMP, or what we call danger or damage-mediated molecular patterns or molecular pathways. So they could be either primed mainly by NF-kappa B signature, which we were not able to find, or they could be directly, people know that they could be activated, and activated by various things, that ATP peroxisomes or LPS in a pathological condition, or in the infectious situation. At the same time, they could be also activated by what we call sterile inflammation, that is induced by DAMP, or damage or danger associated molecular patterns, and which eventually results into release of IL-1 beta from its dormant state, from pro-IL-1 beta to IL-1 beta, and they are also involved in the release of what we call IL-18. So promote the secretion of pro-inflammatory cytokines, IL-1 beta, and IL-18, most extensively studied inflammasome complex are NLRP3 inflammasome complex. We'll focus now on the NLRP3. So one of the key things, or one of the key questions, or the lead what we got from the transcriptome data was that signature of what we call innate immune response. And we have carried out series of experiments to further validate, looking into neutrophil elastase, macrophage, or MPO, that MCP1. These are the various proteins and then enzymes which give you some sense of inflammation if it is occurring. We have carried out extensive cytokine array analysis using quantifying different 50 different cytokines, and we have looked finally into the components of IL-1 beta inflammasome, NLRP3, and release of or the activation of IL-1 mRNA in a time-dependent fashion. So what we, as a whole, we were able to sense that yes, estrial inflammation play, and which leads to activation of inflammasome complex, play a key role in the pathogenesis of hypoxia-induced thromboembolism or hypoxia-induced thrombosis. So to put forward that we had quantified every step, as I mentioned, that inflammasome contains of three major components: NLRP receptors, active caspase, which is converted by ASC2 or adapter protein from procaspase to caspase-1, and then eventually that will release into inflammatory cytokines IL-1 beta and IL-18 release. We looked into all three major components, IL-1 beta, both in terms of protein and in terms of mRNA. Then we looked into caspase mRNA and caspase activity also, and NLRP3 expression pattern. We looked into, as I mentioned, that caspase activity in NLRP3 in terms of protein, IL-1, IL-18 also in protein. So if you follow the red bars, you will be able to see that in case of hypoxic environment, these all were extensively activated or they were hyperactivated. So in the next series of experiment, what we have done, that we got a sense that yes, inflammatory or responses mediated by inflammasome or NLRP3 could result into initiation of thrombus or pathogenesis of thrombus, mainly mediated by IL-1 beta. So what we have done in the next series of experiment, one by one, for a proof of concept, we have inhibited IL-1 beta, inflammasome complex using siRNA approach or silencing shRNA approach, and then you will again know to get into the scientific component of it, but visually also you can see that animals which were in having that reduced level of NLRP3 inflammasome, or that they have a significant reduced clot formation. We have inhibited caspase-1 by using CSC IC or what we call IL-1 beta converting enzyme inhibitors, and then you will again appreciate the similar kind of result we were able to get. And then this all was supported by various coagulation assays, thrombus weight, size, prothrombin, D-dimer, and various other assays we have done. And then third, we have used IL-1 beta neutralizing antibody or IL-1 beta blocking antibody, and you will see that again we have found the similar components. So then we were comfortable that yes, IL-1 beta, that inflammasome NLRP3 is the main key component for the hypoxia-induced thromboembolic disorders, or what we call clotting disorders. So now the second point, the moment you talk of high altitude hypoxia, we wanted to check this, whether this hypoxia is related directly for the activation of this NLRP3 complex. So the first logical point was that whether we will able to inhibit the hypoxic signature by using these all inhibitors, and then we will nicely able to see that if these are chemical K inhibitors from K-mans, so that we were able to see significant reduction in NLRP3 mRNA, IL-1 beta release, prothrombin components, all those given us, and the other hypoxia-induced genes, so gave us a confidence that hypoxic signature remains one of the key things. Now, the moment you talk of hypoxic signature, it comes to one of the key transcription factor or regulator for hypoxia-mediated signaling is that what we call HIF1, hypoxia-inducible factor 1 alpha, which stabilizes in presence of hypoxia, otherwise they are degraded. So I'll just not get into mechanistic even in interest of time. but the key regulators or the key transcriptional regulator or the outcome of hypoxia-induced signaling is HIF1 alpha. So what we have looked into, we wanted to first check that whether this HIF1 alpha as a transcription factor regulates NLRP3 expression, and then we look for what we call availability or the presence of HRE or hypoxia responsive element through which HIF1 alpha binds, and then we were able to nicely doing the chromatin immunoprecipitation assays, able to show that yes, hypoxia, that NLRP3 inflammasome receptor has HRER, hypoxia responsive element binding site, so showing that yes, it binds to, and that the binding is enhanced in the case of thrombosis. The second thing is that we have again done a proof of concept experiment. If we are able to block this or inhibit this HIF1 in the animals, and then you will appreciate that HIF1 alpha, and we again use this siRNA based approach to inhibit HIF1 alpha. And if you look into the third bars, in all, we were able to significantly not only reduce blood clotting time, is that increase clotting time, reduce thrombus weight and size, and then we were able to see a significant reduction in NLRP3 as well as IL-1 beta, both in terms of message data I'm showing and in terms of protein. So what we were able to know as a whole have a clear picture that HIF1 hypoxia-induced HIF1 alpha will regulate NLRP3 expression, which converts pro-caspase into caspase, and which leads to the release of IL-1 beta from its dormant pro-IL-1 beta form. So this has given us the first link to understand inflammasome, the what we call connection between inflammation and coagulation. So again, to cut the story short, we will try to share you. So these are all animal experiments, cell culture experiment, very fancy. We wanted to test this in the actual condition or with for the translational benefit. So what we have done is that we have carried out, series of experiments and collaborated with number of hospitals, and one of the key hospitals from which we got the patient data is the Western Command Hospital, Chandi Mandir, and then Army Hospital RR. Technically, those patients who are getting thrombosis at working at Siachen glaciers would be airlifted to Chandigarh, one of the baseline hospitals, and we have been very thankful for Indian Army to helping us in all possible ways to reaching out to those places and conducting those works. So they have made all resources available to us. So I'll just try to show you one data for both the points. The first story I told you is that the platelets are getting activated in response to hypoxia, this animal studies. And the second is that calpain remains one of the key molecules or one of the key biomarkers or early molecule which triggers thrombosis. So technically, we were able to see to a great joy for us is that yes, P-selectin is a soluble marker for platelet activation. They were significantly high in the patients who developed thrombosis while working at Siachen glaciers. And then the second was that the calpain activity is significantly higher in the patients who developed thrombosis. So the huge joy for converting this our research from bench to bed. And not only this is again a rare moment for Indian science, is that our experiments this protocol was British expedition group which does these kind of experiments or which kind of expedition comes scientific exploration at Chacaltaya Mountains region in Peru. So we shared our protocol, and they were able to see the same kind of pattern in their cohort also. So again, to pride for the country. The second point, the second story which I told you is that we were able to identify that calpain, and before that is that at a messenger level is that inflammasome complex are the key complex which triggers or which induce the prothrombotic milieu, what we call, or which makes the ecosystem for the development of thrombosis. So what again in the another new set of cohort of 30 patients from the control, and then those who did not develop while working at altitude versus the who developed thrombosis while working at CH glacier regions. So believed into all components of NLRP inflammasome, NLRP3, caspase, IL-1 beta, IL-18, the ICE was caspase activity, the release of IL-1 beta, and then the proteins NLRP3. So to again our happiness, this we were able to see whatever we have seen the animal and the cell culture based experiment, we were able to prove that in the patient sample. So that exactly the same kind of, if you remember, COVID is eventually hypoxemia, means that because of viral infection that your lungs get incapacitated, and then that release into what we call cytokine burst, and then that finally triggers clotting. And that's why people were looking into the D-dimer as a test for the severity of the COVID infection. So again, we have carried out this, we have published this work in 2017 and COVID, around 2020. So that gives again great pride for the scientists that whatever you have done earlier has been proven in an entirely different setup, and that could be entirely. So if you remember that the initial phases, there are a lot of these kind of drugs which has been allowed for against cytokine bursts, especially from the CSR labs, also have been approved for this. So this now, as a basic scientist coming to this, we wanted to have in-depth understanding. So till IL-1 beta, we were fine. Eventually, we said that the thrombosis could happen or that clotting could happen. But IL-1 beta is a cytokine. It has to do something for the clotting factor, which will finally make the thrombus or clot. So we have in-depth dealt with the series of experiments. I'll try to show you a few of them using different cell lines. I mentioned few that we have used human, PBMCs, human HUVECs, THP1 cell line, K52, transformed HUVEC cell lines, all possible rat PBMCs, rat platelets, human platelets, to just understand now on the mechanistic level, on the basic science level. And then this again through cartoons, I'll just try to make it a little bit like presentation simpler, is that we wanted to show that one of the key event post hypoxic exposure would be that platelets or the endothelial surface will be activated, and then activation of endothelial cells would lead to the recruitment or adhesion of monocytes on the surface. And again, not to get into rocket science, you can just see the dots here, that hypoxia. And then we have used all chemical inhibitors to stimulators and inhibitors, DMOG as a stimulator to show that chemically mimic that. This is hypoxia again as a cell culture, is a 1% of oxygen, cells grown in 1% oxygen. And then DIM is the inhibitor for HIF1. So technically these all three lines that suggest that yes, hypoxic exposure, if you compare these two versus either control or the DIM, this inhibitor or hypoxia, could result into a higher sticking or adhesion of monocytes to the endothelial surface and that hypoxic environment. In the next series of experiment, we wanted to show, and we have shown, again, not to getting into in the interest of time, I think almost reaching there. So we have looked into all various possible hypoxia-induced gene, VEGF, EPO, erythropoietin, which again is regulated by hypoxic exposure, as well as NLRP3, both in terms of message or in terms of protein release or protein expression. We were able to show that yes, that inflammasome complex in the monocyte that triggers that hypoxia NLRP3 axis, and that is triggered or that is stimulated by adhesion of monocyte to the endothelial surface, and which is highly amplified, 5 to 6 fold under hypoxic environment, which eventually could result into release of IL-1 beta. So still our story was that stuck with, we were able to reach up to IL-1 beta. We wanted to know what IL-1 beta does. And again, to just show you, put the long story short, we were able to identify a new transcription factor, what we call EGR1, and those, you can look into those. They have number of roles, critical role into cancer also, and then EGR blocking antibodies have been used. They are regulated by HIF1 alpha in the tumor cells also, and then in the infectious biology also. So what we have done, what we stumbled upon at that EG early growth regulator 1, and then you will see that they are significantly upregulated in the hypoxic environment, either by the natural hypoxia or the chemically induced hypoxia. And then DIM1 is the inhibitor, and then MCC95 is the inhibitor of NLRP3, and SML is the inhibitor of caspase. So this tells you a complete story that yes, these are activated in terms of hypoxic environment, which are regulated by hypoxia as well as NLRP3 inflammasome complex. We looked then into a gene of EGR1, and we were happy to find what we call again HRE, hypoxia responsive elements. Also, again, this is a chip data to suggest that they are, yes, they have human binding site in their promoter region. But interestingly, what we were able to see is that they are not, they are downstream of EGR, downstream of NLRP3 inflammasome complex. Then we dwelt upon, again to cut short the data, is that what kind of role platelets play in these all EGR expression or EGR regulation. And you will appreciate, again, not to get into so complicated experiment, but what we have done in the cell culture based co-culturing experiment, we have grown these all cells, monocytes, platelets and monocytes on top of endothelial surface. And what we were able to now had a clear picture that yes, EGR1 is regulated by this IL-1 beta, stimulation of IL-1 beta, and which triggers or which activates or modulates another key factor, which is a key component of what we call extrinsic coagulation factor, means the clotting activated by some external molecule or the factors. And one of the key signature for that is the tissue factor. And we were able to see that yes, tissue factor activity is enhanced in the EGR1 stimulated cells. We have carried out experiment by silencing or inhibiting EGR1 both in animals and the cell culture, this complex co-culturing experiment, to come up with the idea that yes, that IL-1 beta regulates EGR or induce EGR expression, which further regulates tissue factor activation. And lately we were able to find out that tissue factor also contains, which in terms of regulation contains SP1 or EGR binding site. I'm not trying to show you the data again to complicate it. So now we have a clear story that hypoxia induced HIF1 alpha activates NLRP3 inflammasome, which further activates or releases procaspases, which converts pro-IL-1 beta to IL-1 beta, and that results into activation of EGR1. EGR1 further binds to tissue factor and then activates or induce that tissue factor activity, which could now we have a clear sense of that tissue factor is a final clotting or triggering event. Again, so what we were able to do with these all series of cell culture or co-culture based experiment is that establish HIF1 alpha, NLRP3, IL-1 beta, and now we have added EGR1 and tissue factor as a critical axis linking hypoxia, inflammation, and finally to coagulation. So again, like our theme of the other works also, we have carried out series of experiments to prove whether this could be, has some experimental thing, has any clinical relevance. And this time, since I was out of DRDO, we have collaborated with various civilian hospitals. And one of the key hospitals in Leh, those who have visited, is that Sonam Norbu Memorial Hospital, SNM Hospital. And there's, again, I'm trying to show you data from, these are my students working with patients. And I have been lucky to get funding from DBT for carrying out all those. So what we were able to, again, to cut down the story, we have taken around 20 samples or 20 patient data, and then you will nicely say that whatever we were able to find in the cell culture based HIF1 alpha also activated, elevated in, stabilized in the patients in NLRP3 inflammasome complex, includes caspase 1, IL-1 beta at the message level, and then interestingly that EGR1 both in terms of protein as well as message level, and then factor free levels, which works in the association with factor 7. So now we had a complete story about we were able to say to, again, not to divert much, is that we have carried out series of other experiments to get involvement, understand the involvement of various other factors, which may not be possible for me to cover all those, but what you will appreciate that 10 years back, we have started just with a simple point that yes, hypoxia induces clotting. Now we have a clear picture, and we are still looking for some, better understanding or deeper understanding of these events. To just make you, that's what make us wow, is that biomarkers, and they're all classical science and they're fine, but what makes us quite fascinating as a scientist is that, again, because of interest of the time, I'll not get into, we have parallelly carried out a huge longitudinal prospective studies, technically followed 1000 soldiers, 1000 volunteers from Delhi to their deployment to the Siachen glacier, and then out of these 1000, that 14 developed thrombosis. So then they have given, it has given us a big window to understand the disease. Now you understand that this is a prospective study where we have all the individuals who did not have thrombosis, and at the end of the study, we have a subclass of the people or sub-cohort who developed thrombosis, and rest of the people who did not. So we have carried out extensive work on a various aspect that has been published in Lancet South Asia. If you are interested, you can look into. They have carried out a nice editorial with it that are efforts to understand the mechanistic, provide the mechanistic evidences into it. And then that parallelly with our team, there was one guy, if you have seen NDTV before, almost like 10, 15 years, Nitin Gokhale was their editor, and who were there for some time with us. He had written a nice book on various aspects, including 2 chapters dedicated toward what we call beyond NG 9842. This is NG 9842 is the name of last base post where India controls the Siachen glacier, their Siachen saga. So if you are more keen to learn about, understand about our fascinating scientific journey, you can look into this book. So to finally conclude that what we have said and how it makes it a breakthrough, or what we have done, is that since we have longitudinal study, you just have to understand, I'm just trying to show one data to finish up my talk, is that we have all individuals at the sea level, which has traveled from Delhi to high altitude to Ladakh and extremely to Siachen glacier. So the red line is that the individuals who developed thrombosis or who became patient at. And now what we have done is that we were able to we went back and looked in our data after this all transcriptomic work, is that whether that we could have seen this signature before. And then now I'm encircled this into for the simplicity. So now what was fascinating is that even before the onset of disease, so EHA is at the stage where they have developed thrombosis. Even before the onset of disease, they were having significantly elevated both IL-1 beta and NLRP3. So that gives a wow factor that yes, either you can use them as a control mechanism or to target them to control the NLRP3 inflammasome, or you can use these cytokines as a screening tool that yes, somehow or other this patient could develop this disease. So that makes us wow, in a sense that yes, we were able to have some tool to detect as well as to target the disease. And then I just that was nicely carried out, this whole story in Nature India. So I'll just stop here, I guess, and then we'll be open to take your questions. I'm by and large right on time, 12:56. So I've been really thankful or supported by various collaborators, both basic science and number of hospitals I'm working with. And then I've been equally blessed to be funding by almost all government funding agencies. But these works have been, carried out extensively by my dedicated PhD scholars. The NLRP3 story I told you about, done by Neha Gupta. Then the IGR story, I said. So this is my DRDO team, and now this is my current Jamia team, was Shankar Chanchal. Then the Tarun Tyagi has carried out campaign work. Now they are very well placed. Tarun is a faculty at Yale, Neha got the Inspire Fellowship and all those Shankar is at NIH, and all my other students have done very well in all the aspects. So I'm equally thankful to them, thankful to all my phenomenal collaborators who had helped me in all through. And then finally, I mean, you are open to ask any questions. You are open to connect me. I am available for not that way very active, but most of the time available for any social media platform if you want to. And then we work on the disease. So one of our major goal of our lab or research is to do the benefit of the society. So I've just shown you hypoxia induced one, but we work on various kind of thrombosis, not only induced by thrombosis. So every year, 13th October is celebrated as a thrombosis day. So we to create an awareness. So please look into this website and look into that who could be more susceptible and how we can minimize this. This remains one of the, after a stroke and MI, that's third major killer of for the mankind. And I've also put in Urdu, सर से पांव तक इख्तियार संभालें, खून के लोथरे से बनने से बचे, अपनी सेहत की हिफाजत करें. So thank you all for patient hearing. I'm dot in time 59. Yes. I'm
Thank you
open
very
to any other questions, anything which you want. I can stop sharing now, and then we'll love to hear from you guys. Yes.
Thank you, sir.
I think we will take the questions once you post it on the messages. So I will be moderating this session. So Avdesh, if you have any question, please post it here. Meanwhile, while you are framing your questions and typing it here,
So a lot of people have audio issues or what?
I don't know, but this might be more convenient. So that's why.
No, I'm saying that a lot of questions are about the audio issue. Was it audible for you all?
That was the inaugural session.
It was audible.
Okay, so there may be their end, a problem at their end. Yes. So if anyone has any questions, for living I do teaching, so I love to talk.
So I have a question.
I have a question. Okay, go ahead. Go ahead.
So because my interest is solely on cancer, but the thing that I learned today is about this NLRP3 inflammasome activation due to hypoxia, which is also found in tumors which are hypoxic at the core.
Absolutely.
And this promotes tumor growth, and it has been involved in metastasis, drug resistance. So how do you see that working this aspect in treatment of the cancer? Have you looked into some kind of mechanism that is common to both thrombosis activation as well as the spread of the cancer?
Okay. So as I've shown you in the beginning itself, there are two connects. The first is that tumor per se is localized hypoxic environment. So multiple things like HIF1, EGF, TLRs, they have both regulated by hypoxia, and they have a big role in cancer. So that's a big area. The other thing is that's emerging as and we are also working on when the cancer meets coagulation, you see. So another thing is that one of the major things and one of the major side effect of chemotherapies are trigger of clotting factors or clotting pathways. So that's another area where the coagulation factors, and they especially now if you look into all three components, hypoxia, clotting factors, and then in our context the outcome was more of thrombosis or clotting per se. But for tumor cells also, they are being found to have these all signatures, and one of the key factors are that what we call tissue factor derived microparticles, which actually entrap the cells and then makes them more vulnerable or more accessible for the clotting factor. So that's a huge area. We have also ventured into one of the, in collaboration with Laval University, Canada, that that hypoxia, how this proofreading could also be compromised, which eventually induce tumor formation is compromised in response to hypoxic conditions. So very nice area to work for, and I mean, we love to collaborate.
I would just expand my question here, because if we look into the neoangiogenesis, because cancer cells, they need new blood supply. And if we are talking about NLRP3 causing thrombosis, which might even block the supply of these nutrients to the cancer, so still they're able to thrive. Isn't
Yes,
that
absolutely. They are parallel kind of studies has been done with the TLR, and the TLR is the kind of, that came into nature, I guess, that TLR is the switch for angiogenesis. In terms of what we study, angiogenesis is loosening of the clot. What in the tumor, what happens is that it strengthens the tumor formation. So they work as a switch to converting this hypoxic or that signature into angiogenic or neoangiogenic signals. So that has been all. So nice to see our Vice Chancellor, ma'am. First of all, congratulations for getting that fellowship, INSA fellowship. It was wonderful hearing you. And then so if you could, ma'am, unmute yourself, will be really all the students will be love to hear from you.
Good afternoon to all of you. I'm
Yes.
sorry I could not make it to be there offline because of two prior commitments which was already I had done, and I'm really sorry for that. I really missed it. The loss is mine. But I wish you all the best, and I'm sure this is going to be one of the best workshop and this thing, and with so many the participants, I mean, joining internationally and from here also the resource persons and all from my side. I'm really thankful to them, and congratulations to the organizers of this GYAN committee. Thank you so much. I'm here. I'll be
Pleasure having you. I think
deliberations.
organizers
I'll be
will thank you
listening
more.
to your deliberations. Okay,
Oh,
thank you so much. Okay.
Thank you very much for joining us, ma'am.
Thank you, ma'am, for
congratulations. Congratulations to all of you. Aziz Saab, as I told you, I could not make it earlier. I just I came back from the other two engagements, and I'm sure this is going to be one of the best.
Thank you so much,
I
ma'am.
mean,
We understand your
interactions.
schedule.
Sure.
you.
I'll be here, and I'll be listening whatever I can understand, because it is not my area. Thank you so much. Thank you so much.
It's our pleasure and blessing to have you. I think organizers has all nice words and praise for your leadership and for the kind of support you provided them to.
No, I'm really happy. Some very meaningful contributions are going to be, and this is going to be beneficial for our students, I'm sure. Congratulations, Aziz Sahab, and all the organizers involved in it. Thank you, all of you. Thank you. Please carry on. You continue, please.
Thank you, ma'am, for joining us.
I'll be online, but without the video. Okay.
Thank you, ma'am. We can understand your commitment. So kind of you that you spare so much time for us. So this is forum. If you are done, otherwise
I think there's a question.
Yeah, please ask. Interact. That's the whole idea. Is not to.
Yeah, there's a
you can
question.
ask any question. No question is a stupid question. You can ask anything. Any.
You want me to read from the message box? Are there platelet-specific genetic variants in North Indian mountain populations that confer resistance to hypoxia-induced clotting?
So genetic variations we have tried to understand. We have spent a lot of effort and energy onto it. Platelet-specific, we were not as a whole. We were not able to make the longest story short. We were not successful in getting any specific variants responsible for making it prothrombotic. Other groups has done. They have seen like Anurag Agarwal has published a paper in translation research with mutation in VEGF and BWF, which makes them prone to thrombosis. But we have seen other way around. We have seen adaptation as a coagulation as a key role in adaptation, and we were able to find, although unpublished data, but we were able to find a key signature of hypercoagulation during adaptation. So yes, it has a genetic role in adaptation, but what our observation or what our finding of the patients are, these are the low-landed individuals who have been exposed to hypoxia. So for developing that, although prothrombotic or thrombophilia profiling we have done, we were not able to see any unique signature. Is it answer his question or?
Yeah, Mohammed,
One line answer is that we were not able to see a clear genetic signature.
Yes, sir. Thank you.
if we have no more questions, we will.
I have a
break.
question.
Yes, sir, please.
Yeah, Professor, you have given a wonderful talk, and I apologize, I just joined a bit late in it. Being a drug hunter, my question is specifically goes on that line. I was just trying to understand. There was a news earlier. A lot of people were working on this target a lot, and being a target, what we have in the form of a caplan, what we find it is most of the people failed. They identify it's a druggable, but the important is it's not a on and off system. Means uh, if you shut down the whole mechanism or inhibit the calpain, so there was a lot of fatality. So, and the biggest issue they found was the selectivity of 1 and 2. So I think you guys are one of the pioneers in working in that. Is there any way you can help industry to find out some selectivity issue to be really dealt with? So that, because Pfizer, Abbott, everybody has worked. They went for a they got a brain penetrant also on these molecules, but somehow they failed it. So it's still no success, no approved drug. They're still struggling. So I think that is one of the scope which you guys being one of the pioneers for that. Is any thought on it?
So absolutely wonderful question. A wonderful comment, I would say, not the question. The first thing is that, yes, we do agree, and there are a number of, this is a common cysteine proteases, correct? So blocking them has a lot of other issues and a lot of off-target effects that has happened. What we have used this discovery as for early screening. Our idea was to, if we are able to detect this onset. We have not looked into targeting because it has been tried in muscular dystrophy, it has been tried in diabetes, and it has so many issues. And then generally for the smarter understanding, until unless we are able to find, let's see, a specific phosphorylation or specific site or something which is required for their dimerization, their therapies won't be that way successful. How we have utilized this finding is that developing an early diagnostic kit. So we have done all the positive predicting and negative predictive analysis, and then we were able to see at least 4 to 5 times more calpain activation. So we have used more on a diagnostic rather than intervention approach. Number one. Number two is that for the first time, these all calpain regulated by the calpain induced or calpain released by activated platelets gave us a clue that not only clotting factors but vasculature, platelet per se, could also be involved in this whole thing. So we have suggested at the initial thing that nobody prescribe aspirin for the stopping clotting. They have for venous thrombosis in terms of, majority of them are for arterial thrombosis where atherosclerosis will come on, where you see the role of platelets. So what we have done and what they have tried is that we have carried out a small aspirin trial where we are able to hold the platelet activation at the lower level using low dose of aspirin. So that has turned out to be indirect benefit of our study. We have used this calpain as an early diagnosis, and then the lead to understand that platelet activation plays a major role for using aspirin as a prophylactic. What you say that it does not have much side effect.
Yeah, absolutely. Being a bit more upstream and ubiquitous across means expression is across. That's you can't deliver a target delivery to the platelet. That could be one of the challenge going forward. Anyway, wonderful to listen, sir. Thank you.
I mean, these are the challenges now. I mean, we can share our numbers and we can continue. Wonderful. So kind of you. Somebody else wants to talk something. Let's see in the chat box.
There is another question.
Yeah, please. The whole idea is the question only. So,
it says, sir, I'm currently pursuing chemical engineering and I'm very interested in life sciences research. Could you please guide me on what core skills I should start developing now and how an undergraduate student can gain meaningful early exposure to biomarker or translational research while linking both fields?
So one of the things what we are trying to do for the engineers is that for engineering background is that we are trying to develop a microfluidics based system or mimic this whole study and whole these kind of systems. So there they can contribute, one of the things. The other thing, the other stories I did not talk is that we have developed various different microRNAs which we are trying to tag with the nanoparticle. The nanoparticle has a specific target or the receptor expressed for there, they will go on the endothelial surface. So we do a lot of few of the bioengineering things also that could be done as a interface with biology and engineering. So those kind of things he can do. He can contact me if he is interested to. We can help him. So we do biomedical sciences, but I did not show that those data at the preliminary stage. We are trying to, I think, Professor Azhar could also be helpful with. He is trying to develop organoids, so those models could be using the 3D based animal cultures, cell culture of the patient derived cells, and where the lot of engineering tools are required. So there he can learn, get the trainings from there also, or he can work on any. Globally, this is a big thing, biomedical engineering, but India is just catching up.
Thank you, sir.
Yeah, sure.
If we don't have any more questions, now it's time for a short break, and let's gather us all after that. Do we have any questions?
We will gather at what time? Can you put up
2 at 2.
2 p.m. Indian Standard Time?
I'll also put that on the chat box. So make sure to join us after the lunch.
Thank you so much for your kind time and listening to our talk.
It was
All the
great,
best for the coming talks.
and you have inspired me so much. So I'm sure it has inspired a lot of young people who are on in this forum. So once again, a big thank you, and we can give a round of applause.
Oh, thank you so much. You have to just share the pizza coupon for lunch.
Yes, I would definitely do that if Gyan allows.
Thank you. Thank you.
Thank you.